Good evening. First of all, let me thank Paula Heller and Mirta Schatten for this kind invitation. It's a great honor to be present at this meeting and share my data with you today. And today I will talk about the model of bone marrow that we have been developing in my lab for the production of platelet. So in my lab, we envision different applications for this model, and you can see a list of some here. So first of all, of course, understanding the physiology of the bone marrow, understanding how hematopoiesis works in a more physiological environment, in a 3D environment. And consequent of this, understanding disease, find a new treatment for the diseases, testing drugs, and in a frame of precision medicine, and I will show you some data about that today. And last but not least, understanding if we will be able to perform a large-scale production of platelets for clinical application in transfusions. So when we start thinking about modeling the bone marrow, we started with the observation that the bone marrow is mainly composed by extracellular matrices. So we have been working for a lot of time trying to understand the role of the different matrices of the bone marrow in the regulation of hematopoiesis in general and platelet production. With particular focus on fibronectin, and Alessandro Malara in my lab is working a lot on the role of fibronectin in directing Megakaryopoiesis and platelet production, not only in the physiological context, but also in myeloproliferative neoplasm. And then different type of collagen, as you can see here, and Vittorio Banante is working on this task, and we have a recent paper that is coming out in Blood Advances demonstrating the importance of collagen 6 in regulating platelet production and functional platelet production. Megakaryocytes do express different receptors for these matrices on their membrane, and this is the first mechanism that regulate the interaction between megakaryocyte and matrices. Keeping this in mind, we wanted to find a material that could be able to be functionalized with these matrices to reconstruct the basic 3D structure of the bone marrow. So we choose the silk from silk-cocoons, and this comes from a collaboration that I started with David Kaplan a long time ago, 15 years ago. We started collaborating together, and we are still collaborating in using this biomaterial to replicate bone marrow. And the reason why I chose the silk is that it's really a fantastic biomaterial that is cytocompatible. We demonstrated that it's not thrombogenic, and so this is the most important feature, because we don't want to produce activated platelets. It can be processed in water, so you don't have to add any chemical to that. And most importantly, you can functionalize the silk with different molecules, and it keeps their activity, it maintains their function, and you can tune the mechanical properties of the silk. And this is very important because we discovered that mechanics and the elasticity and the stiffness of the bone marrow is an important regulator of platelet production and in general hematopoiesis. So when we started producing our model, the first model, I will show you several models in this presentation. We started with these vessels that David was producing in his lab to reproduce the vessel, to use them with like stent. So we took these vessels and we first demonstrated that perfusing platelets in these vessels was not activating them. As you can see here, platelets not only did not adhere to the vessels, but they did not activate. So this is before and after perfusion inside the vessels. Of course, these vessels were porous, and what we did was we reconstruct a spongy structure made of silk in order to reproduce the spongy structure of the bone marrow. And then we were able to develop and to produce different sponges with a different number of vessels inside, with the goal of reproducing the complex network of vessels, inside the bone marrow. And this work has been done mainly by Christian Di Budo, and I will show you many data produced by Christian. And what Christian did is that he took the structure of the sponge and the vessels that were functionalized with different matrices, as I told you before, and he cultured megakaryocyte inside, and the goal was to see megakaryocyte migrating towards the vessels, extending proplatelets, as you can see here in this cartoon, and then we wanted to be able to collect platelets in the vessels, perfusing the vessels. As you can see here, megakaryocytes did migrate towards the vessels. They crossed the vessel wall, functionalized with different matrices, and of course the vessels are porous. And then we were able to visualize and also collect platelets inside the vessels. And as you can see here, proplatelets were going all around the vessel wall as expected in vivo. And then we analyzed the platelets, and first of all, increasing the number of vessels, as I showed you before, we were able to increase the number of collected platelets, and this means that we were able to really mimic the physiology of the bone marrow, and the more network we were able to replicate, the more platelets we were collecting. And these platelets, they looked like normal platelets. As you can see here with the tubulin ring, they expressed, they had granules inside, and they show some functionality in vivo, shape change, adhesion to collagen under flow, and participation to the clot. And from a morphological point of view, proplatelets, they were normal proplatelets inside the 3D bone marrow model, and they showed granules. And as you can see here, also in this picture, the granules resemble the one that we observed in normal peripheral blood platelets from donors. And adding some complexity to the system, we demonstrated that, again, the more physiological we were going with our model, the more platelets we were collecting. So adding endothelial cells to the system inside the vessels, resulting in an increase in platelet collection, but also functionalizing the silk with different protein derived from endothelium had an impact in platelet production. And we have now a paper submitted where we demonstrated that, again, functionalizing silk with different protein derived from endothelium enhanced platelet production. So the vascular niche is really playing a role in directing platelet production in the bone marrow. And then we wanted to simplify our model and also to enhance the capacity of the model to collect platelets. So we thought that we could consider the entire sponge as a bone marrow and the pores of the sponge as the vessels. So we wanted to produce a sponge and a system that was perfusable, completely perfusable. And we did. So again, Christian, he 3D printed a scaffold, a supporting casket that you see here. And this casket was designed to contain a silk sponge, as you can see here, where we seeded megakaryocyte. We demonstrated that the silk sponge was completely perfusable. So as I told you, all the pores of the silk was perfusable. And now we completed also a computational model where we demonstrated that the perfusion is stable and is uniform during all the perfusion and in the entire sponge. And then we were able, as you can see here in this pink drop, to collect again the perfusion at the end and collect platelets. And with not only so an increase of platelet collection, but also we demonstrated that the megakaryocytes were able to adhere to the functionalized silk sponge, the functionalized with matrices, and then they were extending proplatelets towards the pores, towards the hole, and then the flow was passing through the holes, collecting platelets. And again, platelets were similar to normal platelets, as you can see here, tubular ring, expression on CD61, 42B, and some marker of alpha granules. And now these are new results, and these results, this project is carried on by Carolina Miguel. Carolina graduated with Mirta Schattner, and now she's a PhD student in my lab. And what Carolina is doing is, again, adding some complexity to the system. So our idea, our hypothesis is that in the 3D sponge, silk sponge, we can add a different cell type, and we can create an active niche with cells producing their own matrices. And this is driven also by the characteristic of the silk, by the stiffness, the elasticity of the silk. And as you can see here, Carolina first results demonstrated that we are able to populate the entire sponge with stromal cells, and depending on the stiffness of the silk, these stromal cells produce their own matrices. So we are able to create an active niche that can impact on cell function. And most importantly, what this niche is doing is that is maintaining the stemness of the stem cells, the CD34 positive cells. We can see it inside the scaffold. These are cells derived from cord blood, and they populate, as you can see here, the entire sponge. And our goal was, our hypothesis was to think that if we can maintain the stemness of the cells, then we can drive a more uniform and differentiation into megakaryocyte that maybe are able to produce more platelets, also because they can maturate at the same time. And as you can see here, not in 3 weeks, and now we have data also for 4 weeks, we were able to maintain the cells live, but also compared to 2D, we are able to greatly enhance the maintenance of the stemness of the cells. So in 2D, they differentiate after 1 or 2 days. You don't find many stem cells after a few days in 2D, while in 3D we are maintaining CD34 positive cells. And this work is carried on by Carolina with Christian and Alberto Bocconi, who is another PhD student in my lab. And then finally, what we wanted to do, and this is preliminary to the data, I will show you our new data about the precision medicine patient. We wanted to use this system to mimic the phenotype of patient and in terms of platelet production. So if you have a control, as you can see here, you have a certain production of proplatelets inside the silk, and then you can collect a certain number of platelets. If you have a patient that in vivo has an increased number of platelets, such as an essential thrombocythemia, you have an increase in proplatelet formation, and then an increase in the collected platelets. And at the opposite, this was a patient with an advanced grade of myelofibrosis, and he was thrombocytopenic, as you can see here, and we were able to collect less platelets. So with this in mind, we thought that this was a good model to mimic the in vivo situation of. So we first choose the Trombopoyéticos mimetic, and as you know, there are new indications for the use of Trombopoyéticos mimetic in off-label in inherited thrombocytopenias, and we studied inherited thrombocytopenias for a long time, and we chose two different diseases that affect proplatelet production, anchoring LT26 and MYH9. We collaborated with Alessandra Balduini, who was doing a clinical trial to use Eltrombopag for increasing platelet count in patients with these two diseases that had to undergo surgery. So what we did was we performed our experiments in our bone marrow model in parallel with the clinical trial. So we were collecting a blood sample from patients before the treatment, and then we were comparing the outcome of the treatment in vitro and in vivo, knowing that not all the patients treated with Eltrombopag respond to the treatment. So we took our sponge and we did a new model, a smaller model, and again we 3D printed a cascade with two sponges inside to directly compare the treatment on patient cells. And then, of course, we were able to collect platelets and analyze them. As I told you before, again, the stem cells were populating the entire sponge. And this work was carried on by Christian and Paolo Soprano, and another PhD student in my lab. And so we firstly demonstrated that with our system, we were able to test patient cells even if we had a very small amount of blood. So 15 ml of peripheral blood was sufficient to give us enough stem cells, progenitor cells, to test our system. And we demonstrated, as we already knew, that even in our system was confirming was happening in vivo, that there aren't differences between patient and control in terms of output of megakaryocyte, as you can see here, while there is differences both in propleted formation and also in the structure and bifurcation of proplatelets. Then we started to use Eltrombopag. Of course, we use Eltrombopag together with thrombopoietin because in vivo patients, they do have thrombopoietin, and we demonstrated that we didn't see a change in the dimension and diploidy of the megakaryocyte, but of course, with both patients, we saw a change in the output of megakaryocyte in the presence of Eltrombopag compared to thrombopoietin. To thrombopoietin alone. But again, this change was very variable as expected, and we observed in vivo. And going to propleted formation, we saw that in both diseases we had an increase in propleted formation. So the average, we have a median increase of propleted formation, but again, there was a huge variability between patients in both ankyrin 26 and MYH9 diseases. And then we compared our data with the in vivo results. As you can see here, the increase ex vivo compared to the platelet count increase in vivo. And we had a very good correlation between the two data. Demonstrated that with our system, we were able to capture the results and what was happening in vivo, and so to predict the results in vivo. And we also validated the system with iPS derived from patient in collaboration with Anna Raslova. And this was to prove that our system can be used to test drug in general using iPSs that of course allow for perform more experiments and more mechanistic experiments. So our idea in general is that our system is a very valuable system to predict the response to drug before choosing the treatment of patient. So if in this case, if a TPO mimetic doesn't work, we can choose another one without impacting and without exposing the patient to side effects. So with a peripheral blood sample, 15 ml, we can see the progenitor cells in our miniaturized bone marrow model and compare treatment, and then we can analyze proplatelets directly inside the system, and then we can count and analyze platelets and divide responders and non-responders in vivo. And then the same time we can also took iPS derived from patient and test different drugs, the mechanism, and so on, and test not only the response of cells, but also the quality of cells in reproducing the disease. And so this system is a valuable system for clinical application and also for research application and banking of cells that really reproduce the disease. So in conclusion, the key concepts of my presentation are that the silk bone marrow is a valuable system, and it provides is a versatile tool for studying hematopoiesis in general, megakaryopoiesis and platelet production, and it really reproduces the future of the bone marrow. And now we are adding complexity, and adding complexity, we are demonstrating that we can really reproduce the function of the bone marrow. It's a useful tool to study disease, and it's a useful tool to study drug and to predict the response of drugs in patient. With this, I want to thank all the components of my lab that you can see here, and I already mentioned some of them that participated to the project, my collaborators to this project, of course the funding. And I thank you very much for your attention, and again, Paola and Mirta for this invitation, and I'm ready to take your question. Thanks.